We will be starting the session with Professor Steve Lutz's talk on multivariate statistical analysis, MSA, and classification of particles. Before we start, I just want to request people to come here by 9:20 from tomorrow, because we can start the session on time 9:30. This is not only timed here because people, some people might be online also waiting for us. Okay, so please come at 9:20 so that we can gather and then we can start exactly at 9:30 from tomorrow. Thank you. Steve.
Morning. If you're getting worn out now, just imagine how you're going to feel after another 7 or 8 days. All right, so this is my third and final talk of the workshop. Today we're going to try and talk a little bit about particle classification, and I'll start out easy with some examples, and then we'll get into a little bit of math again, but hopefully not too terrible. So there are two general types of classification that you do with single particle data. There's so-called supervised classification, and there's unsupervised classification. The difference between those two is supervised classification is based on references. So in other words, you're classifying particles into groups defined by existing three-dimensional or two-dimensional pictures or three-dimensional maps. Unsupervised classification is so-called reference-free classification, meaning you're comparing the particles to each other and you're trying to group them together based on their mutual similarity, but you're not making any assumptions about the underlying structure that's involved. So when you start in a project, you're typically beginning with unsupervised classification, and then you're gradually moving towards supervised classification. I'm going to start with the supervised classification here, just because it's the easier category, and you're going to be hearing other talks later in the week which are basically on this topic anyway. So I'm just going to use this as an opportunity to go through the how EMAN does refinements slides really quickly. And then you'll hear more later in the week about how other strategies work for doing basically the same task. So in a normal EMAN refinement, which can be related to some of these other refinements pretty easily, we start out with some sort of an initial model in 3D, and we start out with a whole bunch of particles. Obviously, I'm only showing you 16 of maybe 10,000 or 100,000 or a million particles. And the initial 3D model doesn't necessarily have to be very good. In some cases, it needs to be good. In other cases, it doesn't need to be good. Generally, in EMAN, the initial models don't matter too much, although there's some caveats to that statement. So the idea is we're going to use the initial model as a way of determining the orientations of our individual particles. And that's basically a classification problem. So we make uniform projections of our initial model in all different orientations. Obviously, that's more than 9 orientations. I'm just, again, showing you a few. And because my initial model here is just a Gaussian blob in space, my projections don't look very interesting. They are slightly different from one another. This one's kind of round, and this one's sort of stretched out vertically, but there's really not very much difference at the beginning of this process. So we then take every particle and compare it to the set of projections and figure out which one it looks the most like. So that's a classification process. So we wind up with for each projection in each orientation, we have a set of particles associated with that projection rather than being associated with one of the other projections. And again, of course, there's more than 3 in each of these classes. I'm just showing representative examples. Now, in this first stage of the process, when we have a poor reference, this obviously doesn't look very good. Once we have the particles classified, we then go through this iterative process to align the particles to each other and create a class average. Now, this is one step where EMAN differs from packages like Spider or FreeAlign. In those packages, you just determine the particle orientations and build a 3D model directly from the particles. In this case, we add this additional step of demanding that the particles are self-consistent. So instead of just aligning the particles to a reference and saying, okay, that's where they go, we're going to align the particles to each other and make sure that what the average you get, the thing that you're putting in the 3D map, looks like the particles rather than like the reference that you started with. So we do this class averaging process. So you can see now that the class averages we get look considerably different than the projection which was used to generate them in this particular case. They still don't look very good, but at least they look more like the structure that we're after than the initial model did. The structure we're using, this little quick example, is Growy Owl because these slides date back to 2002 or something like that. So we then take all of these class averages we've got, and again we've got one class average for each projection, and we build a 3D model based on those. Now we have a 3D model which looks more like the particle, hopefully, than the model that we started with, and we can make projections of these and then go through this process again. And after we've iterated that process a few times, it will hopefully converge to the correct structure. Now, there's no absolute mathematical guarantee that this process will converge to the right structure in all cases. This is sort of like an energy landscape, and there can be local minima in the energy landscape, and you could get trapped in a local minima, and that would be the wrong structure. Now, the nice thing is, in reality, the local minima aren't very common when you use this particular, the energy landscape defined by this particular process. So for each structure, there may be 2 or 3 false, sort of local minima that you could get trapped in if you use a particularly bad initial model. But in most cases, you can just start with random initial model and eventually wind up in the right answer. From when we were doing the example on the first day and we did our initial model generation, the initial model generation is basically using the same process. It's just that we're only giving it a handful of pre-existing class averages to use to make this, to go through this iterative process and produce a 3D map. The more particles we give that process, the more class averages we give that process, the more likely it is to converge to the correct answer. And the less we downsample the data, the more details there are present, the less likely that they'll get trapped in some sort of a local minima as well. When we do the heavy downsampling and the heavy reduction in the number of averages, it's really just for speed, not for accuracy purposes. Okay, so when we have the, you can see I was in China recently. So when we have the projections, the class averages basically represent projections of the 3-dimensional map. We then need to take those and reconstruct them. Again, you're going to hear a whole lecture on this topic later in the week, but the basic idea in EMAN is we do this in Fourier space. And in Fourier space, a projection corresponds to a slice going through the origin in Fourier space. For those of you who know X-ray crystallography, this should be a very familiar concept because it's the same concept of determining all of the X-ray crystal scattering peaks. You rotate your crystal in the beam, and you figure out 3-dimensionally where all these peaks are, and then you put the peaks into Fourier space, and then you invert the Fourier transform, and you get your map out. So we're doing something very similar here. We have our class average. We know its orientation, so we insert this slice in Fourier space. Then we insert more slices in Fourier space and more slices in Fourier space, and eventually we've filled up this Fourier volume, and we invert the Fourier transform, and we get our structure out. Okay, so there are variations on this process. Different software packages do things a little bit differently. So a typical process in a MAGIC, for example, would involve a process of unsupervised classification. The projections would only be used as alignment references, and then you determine the orientations directly from the particles without necessarily referring to the 3D model. I'm not saying that's how a MAGIC always works. I'm just saying that that's a strategy that can be used when you do refinements in a MAGIC. A MAGIC, Spider, these packages are very generic, and you can do all sorts of different things within them. A strategy that would be used in a package like FreeAlign or Spider is a much more straightforward process. A more straightforward process where all you're doing is you're determining the orientations of the particles by reference to the projections. So you compare each particle to each projection, and then you say, this particle is in this 3D orientation, and then I just directly put the particle into the 3D map. The problem with doing things that way is a problem called model bias. Even if the particle isn't very good, you can still get an alignment for that particle, and you can end up forcing the 3D map to look more like the initial model that you started with. So the process converges much more gradually. So if you're doing a refinement in a package like FreeAlign or Spider, typically you want to do refinements for 30, 40, 50, 60 iterations before the process converges. In EMAN, since we have this iterative class averaging process here, where we make the class averages look like the particles, it actually converges very quickly, and usually it only takes 3 or 4 iterations before the process has converged. Okay. So that's supervised classification. That's all I'm going to say about that today, and I think you have whole talks later in the week on determining particle orientations and that sort of thing, so I think we don't have to belabor that point anymore. So unsupervised classification is the process that we do when we don't know what we've got. We've got a whole bunch of particle images, and we're trying to figure out what's going on with them, so we want to group them together to get rid of some of the noise and see them better. So for this little discussion, I'm going to use one of my favorite examples, I guess I should say one of my favorite unpublished examples, which is fatty acid synthase. So this was a project which we worked on a decade ago, and another group was working on it about the same time, and they published results which were kind of similar to what I'm going to show you here today, published them first, and we just kind of lost interest and never actually published it, but it makes a really good example for dynamics. So, you know, we'll still talk about it. So fatty acid synthase is a molecule that's present in basically all cellular systems because it's responsible for synthesizing the hydrocarbon chains that go into lipid molecules, so everyone needs to have those. The form of the fatty acid synthase is very different in different organisms. In a lot of lower life forms, you know, bacterial systems, it's a whole bunch of independent enzymes which just float around in solution. In most eukaryotic cells, however, it forms these enzyme assemblies. So a single polypeptide backbone has multiple enzymatic activities. They're all tethered together into one big structure. In the case of humans and birds and a few other organisms, these are Homo Dimers. Okay? So each of the monomers has 7 different enzymatic activities in one polypeptide chain, and 2 of those get together to form the overall structure. Obviously, since it's involved in lipid production, it has a strong role in obesity, it has a strong role in cancer, they're highly expressed in a lot of tumors, so they're very important proteins. Now, the protein was first discovered in the late 60s, early 70s, something like that, long time ago. From the first day it was discovered, people were trying to crystallize it. It wasn't until 2006, I think, that the Ban Lab finally managed to grow a semi-decent crystal of this thing. The resolution is still only 3.2 angstroms. And this was after many, many struggles and many, many years. So it's an okay crystal structure, but it's not a perfect crystal structure. That's what it looks like. This is a map of where the different enzymatic activities are in the structure. Okay, so we have a sequence of 3, another sequence of 3, and then at the very end we have this TE, which basically you have it you seed it with a 2 carbon chain, and then every time it goes through this 6 cycle, it adds 2 more carbons to the chain. Once it reaches the appropriate length, the TE domain here chops it off and it dumps the hydrocarbon chain out into solution. Okay, so it basically just goes through this process over and over again, reaches some length, dumps it into solution, then it starts again from scratch and keeps going. Now, the specific example I'm going to be showing you right here, the TE domain has been removed, and our idea in doing that was we were trying to tether it so it was less flexible in solution. So it would basically build a hydrocarbon chain up to its maximum length, and it would stall, and it would have this hydrocarbon chain tethering it in one position somewhat. And it kind of worked, but anyway, the enzyme we're going to be looking at only has 6 enzymatic activities. Now, there's a problem here. If we look at this process number 3, the distance from here to here is 80 angstroms. Now, it turns out that one of these domains, right here, this acyl carrier domain, so the substrate basically sits on the end of this little tail, which is sticking out in space. And it's able to dangle around pretty freely, but it's only 20 angstroms long. So how does a substrate on a 20 angstrom dangling tail span an 80 angstrom gap in space? So this was a puzzle for a very long time. So this is what happens when you look at cryo-EM average of this. So we collected a bunch of data, we did 2D classification, and this is an average of the top view. And you can see it looks very much like the crystal structure, so you say, okay, everything is self-consistent. We still haven't really answered the question. Okay, so this is what the particles look like. You can see they're kind of noisy looking. It's hard to see a whole lot present in them. So we want to have a process to take a set of 100,000 images like this and then generate 2D class averages from them without any bias. So this is the outline of the overall process. So we start out, now unfortunately the particles that I'm showing you here are actually from a different example. This is a GROEL half ring with associated GROES. Don't worry about that too much. This is just a schematic example of how the process works. So we start out with a set of particles. We start out with a set of references, and the references are just 2D references. They have nothing to do with any sort of 3D structure. I mean, you could use something from a 3D structure, but you don't need to. So we take the particles and we align them to the references. Okay? Now, this is not a classification process. We're not saying this particle looks the most like this particle, put it in that class. What we're saying is this particle looks the most like this particle, put it in that orientation. So the goal in this process is not to classify the particles. The goal is to get the particles into a self-consistent set of orientations. Now, obviously, you can't say that a view like this and a view like this have the same orientation. They don't look the same. They're not the same particle. So all we're trying to do is we're trying to ensure that if the particle looks like this, it's always in this orientation. If the particle looks like this, it's always in this orientation. So we're just trying for some level of self-consistency. Once they have been aligned, we then perform this process known as principal component analysis, which we're going to be talking about very soon, which allows us to classify the particles. We do PCA, we then project the aligned particles into what's called a PCA subspace, and that allows us to use another algorithm called k-means to classify the particles in different groups, and then we can do the same process we used in 3D. We can align the particles to each other and make class averages. Once we have the class averages, we then go back here and we use the new class averages as references, and we iterate this process a bunch of times until we have a nice self-consistent set of class averages, and eventually we'll have something that looks pretty good. And you can see here, this is already sort of the final answer. We see from this mixed set of particles, we can see a whole bunch of different orientations and a bunch of different associations. You know, this has the GROES ligand on it. This one doesn't have the GROES ligand on it. That sort of thing. So if we do that process for fatty acid synthase, we see something like this. And the little numbers you probably can't read in the corner are the number of particles that are in each class. It's around 1,000, so this is 1,076, 1,438, 690. So each of these class averages we're looking at here have a lot of particles in them. Okay? As I said, the data set we started with here was 100,000 particles strong. Now, if we look carefully at these class averages, you'll see something interesting. So we see this view that I showed you before, this sort of top view of the particle, and it looks like it looked like before, but we also see stuff like this. Now, the question is, when I see this, am I seeing a different conformation? Do I have this like this? I mean, is there a rotation occurring? Or is what I'm seeing just some funny rotational change? You know, maybe if I take this and I put it in a different orientation, it actually winds up looking like that. Now, to belie that possibility, if we look, for example, at this domain here and here, that particular domain looks pretty much the same in those two images. There's some changes up here, but it really doesn't look like there's very much rotation, if there's any rotation at all. So we're going to do another classification process to try and sort this out a little better. So what I'm going to do is I'm going to find all the class averages which have a self-consistent lower domain here, and I'm going to pull out the particles associated with all of those classes, and I'm going to make a new data set from that. And that new data set's only going to have, you know, maybe 10,000 particles or 5,000 particles in it. It's still going to encompass maybe a little bit of orientation change, but it'll mostly encompass conformational change because I have one domain which I'm defining as basically being the same in all of these images. Okay? So then I do the classification process again on that subset of particles. Okay? Oh, this is the process I'm describing, right? So we did the reference-free classification, we found particles in a similar view, and now we do another reference-free classification on the new subset. Once we've done that, and I'll show you the results in the next slide, we sort all of the class averages we get from this new process into order based on mutual similarity. So what we're looking at here is basically a population dynamics study. Okay, we're not looking at one particle and how it's changing over time. We can't do that because as soon as we hit it with radiation in the electron beam, we're destroying it. But we can look at the whole population of particles and say, well, this particle looks very similar to this particle, but it's bent a little bit. This particle looks very similar too, but it's bent a little bit more. And we can string things together into different orders based on different apparent motions that we see. So we can give you the perception of motion where it's really just population dynamics. So then we can make a movie and look at the results. Okay, so this is what we get when we do that analysis. And you can see there's actually quite a lot of interesting stuff going on here. In all of these images, basically this lower domain looks pretty much the same. There's some slight changes in a few places, but it's pretty much the same. But we see all sorts of funky rotations and rocking motions and stuff. So what happens when we string this together into a movie? This is actually a pretty long movie because there were a lot of class averages. I didn't show you all of them before. So we can see there's a whole bunch of things going on here. There's a rocking motion like this, where the top and the bottom are tilting with respect to one another. If you look at the bottom, in one point in this movie, you'll actually see the bottom domain twisting out of plane, like this with respect to the top. And we also did a study on the unmutated form of the enzyme with the TE present, where it wasn't tethered together. And if you have that domain, you can actually see the bottom domain rotating almost 90 degrees with respect to the upper domain. Okay? If you look carefully here, you'll see something that looks kind of like it's just randomly bouncing around in space. That's where the TE domain is. That's that arm that's sticking out and carrying the substrate around. So what we're seeing here, we're focusing the ordering of this based on the overall rocking motion of the protein. If instead we focused on this domain and we resorted the movie, what we would see is ordered motion of this TE domain where the rocking motion was just bouncing around randomly in space. Now, if those two motions were coordinated motions, if the motion of the TE domain only happened in a certain way when the rocking motion happened a certain way, then it wouldn't matter what we sorted based on. It wouldn't matter what feature we focused on. Then we'd always see sort of one self-consistent path. What we're seeing here is that we have a bunch of different independent motions going on in this enzyme. So it's basically driven by just random motion in solution, rather than a specific guided pathway. But this tells us now, finally, how the substrate is able to visit the site over here. It's bending and twisting such that the TE domain is getting the acyl carrier domain is getting close enough to the different enzymatic activities that it can actually visit all of them. Now, you might say that still seems like a pretty inefficient process. Wouldn't it be better if it was sort of forced to visit site 1, then site 2, then site 3? I mean, if there was some physical process which forced it to happen in order, it's probably true. But if you remember, I mentioned in bacterial systems, these enzymes are actually just different independent free-floating enzymes in solution. So if you consider the relative efficiency of the enzymatic system, if it's floating freely in solution, it's going to have to wander around for quite a while before it finds the next enzyme in its lengthening process. Whereas in this case, at least all of the enzymes are in proximity to one another. So the chances that it's going to encounter an enzyme soon, the next enzyme that it needs sooner is much higher in this case, even if the process is somewhat random. Okay, so that's just one example of how you can do it. We can do other examples of these sort of dynamics movies as well. This is IP3 receptor, where we can see some motions in some of these domains. So fatty acid synthase has these really big sort of 30 or 40 angstrom motions. IP3 receptor has some smaller local sort of 15 angstrom motions. The trick, which is a chaperonin, has these more localized sort of 12 angstrom motions up here in the apical domains up here. But all of these particles undergo some sort of motion in solution. Now, these are all in states where we didn't try and sort of lock them down in one biochemical state. So if you're trying to do a higher resolution structure, obviously you want to biochemically control the situation, so hopefully things aren't moving around too much. But I would tend to view this as more of an opportunity than a deficiency in the technique. There's no other way that you can really gain access to this sort of level of structural dynamics information, right? You could compare this sort of the results like this with molecular dynamics, and then you could actually directly sort of see what's going on in this sort of thing. Whereas if you're just solving a crystal structure of something, you see one state. You could soak the crystal and get it into second state maybe, and then you can see another state. but you really won't be able to see this sort of continuous motion that's going on here. So even if this doesn't lead to high resolution, it can lead to a lot of useful information. Okay, now that was the last slide in my talk that you really have to understand to sort of make use of these algorithms in practice. The rest of the talk is going to be a little bit more esoteric again, and you don't have to understand it, but I think it's actually really interesting to understand a little bit about what's going on here. There's a certain mathematical beauty to some of the stuff that we're going to talk about here that if you understand it, it can give you a sort of a different way of looking at things. But if you don't understand it the first time you see it, don't panic. Most people, I mean, with Fourier transforms, the first time I saw a mathematician present Fourier transforms, they just kind of went, what in the hell? You're doing this big integral of sine waves and stuff like that. It was done from a mathematics perspective, and he was talking about all of these obscura. You could use it to solve differential equations and stuff. And I'm, why? What are you doing? Right? You see it another time, and you see an electrical engineering professor present it. Then you see it another time, and you see a physicist present it, and they all present it from different perspectives. And eventually, you start understanding how these things work. The same thing is true of some of this. So if you don't completely get it today, I'm not going to be offended that I didn't teach it well. Okay, so we're going to start with something very simple, orthogonality. Okay, so we have a pair of xy axes here. They're unit axes, unit vectors here. So x goes from 0 to 1, y goes from 0 to 1 here, and I'm going to plop a vector down in space. Okay, so this vector is here. How do we specify the coordinates of the vector? Well, we take a projection of the vector onto the x-axis, and we take a projection of the vector onto the y-axis, and then that tells us that this vector has the coordinates 0.8, 0.5. That's the vector. Okay, what happens if we rotate the coordinate system? And I put the same vector in the rotated coordinate system. Well, in the rotated coordinate system, I can do the same thing. I can take projections along the x vector and a projection along the y vector, and now my vector, it's the same vector. I didn't do anything to it, I only changed the coordinate system, is 0.2, 0.9. Okay? Okay, so the way we do that is, the way we do that rotation operation is through what's known as a rotation matrix. So this is a two-dimensional rotation matrix. So I have some vector, x, y, and I multiply it by this 2 by 2 matrix, and I get x prime, y prime, which are the coordinates in the rotated coordinate system. And I have one variable here, theta, which tells me how much I rotated the coordinate system. Okay? Very simple vector mathematics. If we go, so we can extend this concept into multiple dimensions. Right? So if we have two independent values, that corresponds to a vector in two-dimensional space. If we have three independent values, that's a point in three-dimensional or a vector in three-dimensional space. And I can have n independent values. So my input vector could have n components, and my output vector would also have n components. And I can do all these rotations. So a 2D rotation matrix has one angle to specify. If we go to 3 dimensions, though, there are now 3 angles to specify. Right? Because I can do I can rotate in xy, I can rotate in xz, I can rotate in yz. I have to specify 3 angles to uniquely define the orientation. So these are the sort of the Euler angles that you're getting familiar with anyway. When you do cryo-EM single particle reconstructions, each particle has a set of an orientation. That orientation is expressed in terms of these 3 Euler angles. We can also go to 4 dimensions. Now this isn't one whole matrix because it's just too big and too messy. So what I'm showing you is the xy matrix and the yz matrix and the xz matrix. So there are 6 different rotations that you have in 4-dimensional space. Now we went from 1 to 3 to 6. So there's a general formula. It's n times n minus 1 over 2. So it more or less goes as n squared. So as the dimensionality increases, the number of different angles we have scales very quickly. So we're talking about a 100-dimensional space. That's quite a lot of different angles that we could ostensibly specify. Now, why in the heck would we want to do rotations in a 100-dimensional space? That's a good question. Okay, so let's look at an n-dimensional real space basis. So what do I mean by a real space basis? So this is just a diagonal matrix with 1. So this is basically saying that we're not doing anything, right? This is a unit matrix. It's 1 along the x-axis, 1 along the y-axis, 1 along the z-axis. So, Yeah, so this axis corresponds to x, y, z, and x prime, y prime, z prime, et cetera, are all exactly the same as they started. Okay, now the rotation that I was just doing was one example of what's known as an orthonormal transform. Say orthonormal has got two parts to it. There's orthogonality and there's normality. So an orthonormal transform is a transform where if you have a set of vectors in the space and you perform the transform, all the vectors will have the same relative orientation to one another after you do the transform, and they'll all have the same length. They won't get longer, they won't get shorter, that sort of thing. You can do arbitrary transforms where things get longer and shorter and they get stretched and mushed and stuff like that. Under an orthonormal transform, which are things like rotation, everything is the same, it's just in a different orientation in space. So the things you can do when you want to do an orthonormal transformation are rotation, reflection, and permutation of axes. So I could swap the x-axis with the y-axis, and that would still maintain orthonormality. Or I could mirror things across the xy plane, and that would preserve orthonormality. Or I can do rotations. Now the nice thing about these sort of transforms is, if I want to invert the matrix, so if I say I'm going from my original coordinate system and then I'm doing a rotation to go to the new coordinate system, if I want to invert that process, I just need to rotate in the opposite direction. And rotating the opposite direction corresponds to taking the transformation matrix and just doing a transpose of the matrix. You just flip the matrix over, and then that will undo the transformation that you just did. And if it's an orthonormal transformation, that's always going to work fine. It's always a reversible process because you're not modifying anything. Okay, so back to our real space basis, where we are, our x prime, y prime, and z prime are here. If we look at this matrix, the rows in the matrix correspond to our unit vectors. So if we think about just a two-dimensional space where we have this 2 by 2 matrix here, this is 1 along the x-axis and 0 along the y-axis. Then it's 0 along the x-axis and 1 along the y-axis. So this is one of my unit vectors, and this is another unit vector. If I looked at my 2D, I think I may have, no, I don't have it here. I'm going to go back here for a second. If I look at my 2D rotation matrix, this is one vector in my rotated coordinate system, and this is the other vector in my rotated coordinate system. Okay? So the rows in the matrix correspond to the unit vectors that we're using to figure out the projections of things. All right. Now here's an example of another matrix. This one is a non-orthogonal matrix. So in this case, each of our unit vectors has 3 components in it, and the components overlap. So if you look at what this is doing, is it's basically doing kind of a local convolution. So if I put in a vector, it's going to combine the values, in this case, it'll combine the x, y, and z values into the new y value. And here it will combine the y, z, and w values into the new z coordinate. So it's combining things in such a way that it doesn't preserve this orthonormality property. If I need to take the inverse of this matrix, I need to undo that convolution operation that I'm doing. The matrix that I get back out is definitely not the transpose, it's this horrible, messy thing. And the more convolution I do, the messier it gets. Now, when we talked about convolution and deconvolution with Fourier transforms the other day, we found a general way of doing this without having to do complicated matrix math, but it's the same basic concept. All I'm doing here is a box convolution function, and then I'm undoing it with the inverse form of the matrix. All right. And the square root of 1 over the square root of 3 here is just a normalization thing, so the lengths are still preserved. Even though this is not orthogonal, it's still normal. All right, now I want to take you back to Fourier transforms from a few days ago for a minute. Okay, so this was the final form of the finite discrete Fourier transform that we looked at the other day. And note that k, x, and n here are all integers, okay? And that they have limited range. So again, f of x was our input function. In this case, it's discretely sampled. So if I have 32 samples that I put in, I get the 16 sine and cosine coefficients that come out on the other side. So x and k are the same size vectors. And then I have my e to the minus I 2 pi kx. The 2 pi here is just to make sure that our oscillations in the box always start at the beginning of the box, and a full oscillation ends at the end of the box. So it's basically e to the minus I kx, and then there's a 2 pi over n here. And then we're going through all of the different n's, which are the increasingly small oscillations inside the box. Okay, so what I have here is an input vector and an output vector, and then I have some individual component vector that I'm multiplying by here, this e to the minus I2kx. So I can look at what that looks like. So here's the first unit, the first vector in my FFT basis. This is just the constant one, the trivial one at the beginning. Here's the first real one. Okay? So we've got this one oscillation in the box sine wave. Now we've got two oscillations in the sine wave, three oscillations in the sine wave. And as they get shorter and shorter periodicity, they look less and less like sine waves, but they're still following a sort of sinusoidal pattern. When we get all the way to the end, it's just this 1 minus 1, 1 minus 1, 1 minus 1 thing, just like we talked about the other day. So these are the basis vectors for a Fourier transform operation with 16 components in it. Okay. Now, the thing that may not be obvious from looking at this is that all of these basis vectors that we're looking at here, all of these sets of 16 values, are orthogonal to one another. What that means is, if I take any one of those, if I take this one and I multiply it by this one and sum up the result, I always get exactly 0. The dot product between any two of these, this one and that one, any two of these I can multiply by one another and I always get exactly 0. The generalization of that statement is just this, which is if you take a sine wave and you multiply it by another sine wave of a different frequency, what you get out is a pair of sine waves at higher frequency. They're still sine waves. And if you integrate a sine wave over an exact a unit number of periods, well, you get 0, right? A sine wave integrated over one cycle, it's got an equal amount of positive and an equal amount of negative, so it always comes out as 0, right? So that gives us this nice orthonormality arrangement, as long as the sine waves or cosine waves that we're using have an equal number of periods inside the box. So even though we are doing this funny sampling here where it doesn't look very sinusoidal anymore, it still holds. Uh-oh. Apparently I need to log into my Wi-Fi. Okay, so what that means is we can now take our Fourier transform and we can write it as a matrix operation. So this is a 16-element Fourier transform matrix. If you take a 16-element data set, you know, a 16-pixel image, and you put it in as a vector on the one side, and you multiply it by this matrix, what you get out on the other side is the Fourier transform. Okay? It's kind of it's a very simple concept, but it's also kind of weird. The reason it's weird is because what I'm saying is that all these input vectors are orthogonal to one another, and we were just talking about rotations, right? We're talking about what orthonormal transforms are. What that means is, in a certain sense, a Fourier transform is just a really weird rotation in a very high-dimensional space. Okay? So a Fourier transform, we're saying, oh, you're multiplying this by sines and cosines, I'm doing all sorts of funny things. Really, it's just a rotation, maybe with some axis flips or something, but it's really just this weird high-dimensional rotation. Well, that might lead you to start thinking, maybe, you know, I'm in 16-dimensional space here. If I've got this Fourier transform matrix here, maybe there are other things I can do too. Maybe there are other funny rotations I can do which might give me interesting results. Certainly Fourier transforms are very useful. Maybe there are other kinds of transforms too, and of course there are. So real space is, of course, already a basis. I mean, it's already a set of basis vectors which are useful. So when you have real space, you get very accurate position information. You know that this pixel is here, it has this value in this particular location, but you don't get any information about the spectrum, the power spectrum. You don't know if there's a strong component of one particular frequency present in the data. You only know where the things are. And the Fourier transform is the opposite. In the Fourier transform, you have frequency information, but you don't know where in the box that frequency information exists unless you take it back to real space and look at it, right? So these two spaces are sort of dual spaces which serve these two distinct purposes. Now there's something kind of halfway in between called wavelets. You may have heard of wavelets before, even if you don't know what they are. So wavelets are kind of halfway between a Fourier transform and real space. In a wavelet, you do something that looks kind of like a sine wave, but it's localized in space. And they don't look exactly like sine waves. I'm not going to talk about wavelets in detail today. That would be another hour-long talk. But wavelets give you kind of a mix of location information and frequency information. Now, the thing we're going to talk about next is principal component analysis, singular value decomposition. There's different names for it. And it turns out that that also is forms an orthonormal basis, and a very useful orthonormal basis, which we can use to classify particles and we can use to get rid of noise and that sort of thing. There are many others as well, which I'm not talking about. Okay, so there's three terms which you often hear used somewhat interchangeably, incorrectly, in the cryo-EM community. So there's MSA, PCA, and SVD. So MSA is just a generic multivariate statistical analysis. So it's statistics performed on large numbers of variables. So if you think about having a 100 by 100 image that has 10,000 pixels in it, that each of those pixels is a variable. So one image corresponds to a 10,000 variable single sample. I have another image that's another 10,000 single samples. So when I talk about multivariate, it's the 10,000 pixels in the image. When I say statistical, that means I have a whole bunch of images to analyze. So single particle analysis is clearly in the realm where multivariate statistical analysis is appropriate. But MSA is a large category of algorithms. It's anything that you might use to analyze that sort of situation where you have a lot of variables and you can do statistics. PCA, principal component analysis, is a specific type of multivariate statistical analysis, and it's designed to look for, as we'll see very soon, the strongest information content in the space defined by these very high-dimensional objects. Then finally, singular value decomposition is a specific algorithm that you can use to compute PCA. Singular value decomposition also used for other purposes in linear algebra and matrix math and that sort of thing, but one of the things that it produces is the result, the PCA results. Now, in practice, we don't actually use this algorithm to do it. There are other algorithms for doing PCA which are much more efficient, but you can do it with singular value decomposition. Now, I haven't really explained what PCA is yet, so we need to move on to that. So we're going to start with something really simple: 2-pixel images. Okay, so here are 6 different 2-pixel images. We are then going to label the first pixel as X and the second pixel as Y, and then we can plot all of the values. Now, when I plot all the values for these 6 images, I'll notice that they all happen to lie along this line. So there's a very strong correlation, in fact, in this case, a perfect correlation between the value of this pixel and this pixel. Now, if I think about how I want to represent this information in the most compact possible way, it's kind of silly for me to have these two independent variables here. In reality, I only have one variable, where the pixel pair is along this line. So what I would do is I would fit a line through the data. I would fit a line through the data, and I could put in another orthogonal line, and then I can rotate my coordinate system based on those 2 vectors I just defined. And now my x-axis defines all of the information I ever need to have about that pair of pixel values. There's no information in y. The pixels aren't deviating in y at all, so there's no information there at all. Okay, so now after my transformation, after going through this matrix, I can take my x value and that if I have one value for a value of the first pixel, I know the value of the second pixel exactly. All right, what happens if we add noise to the system? Sorry, this is the same thing. So this is just saying this is an orthonormal basis, which hopefully is pretty obvious. Okay, so if I add some noise to the system, again, I still have a very strong correlation along this particular axis. So I can still fit a line through this axis, and I can say that the strongest correlation direction is going to be my primary axis, and I'll go to make an orthogonal axis to that primary axis, and that'll be my secondary axis. So I can do the same transformation that I was doing before. It's just that now my y-axis in the rotated coordinate system isn't 0 anymore. Now there's a small value associated with it. Now there may be a correlation in that value, or it may just be completely random noise. If I say all of my information is contained in the x-coordinate, and I ignore the y-coordinate, what I've done is I've actually gotten rid of a lot of the noise that's present in this set of data. I haven't got rid of all the noise. There's some noise which actually exists along this direction as well, but some of the noise is in the orthogonal direction, and if I know that that's pure noise, that there's no information contained there, I can eliminate it, and I can thus eliminate some of the noise. and this is kind of the basic idea behind principal component analysis. We look for strong correlations that may be present in the data, and we focus on those strong correlations, and we ignore everything else. So if we have 3-pixel images, then we go into 3-dimensional space instead of 2-dimensional space. And in that situation, it wouldn't just be correlations along x and y. I could have correlations along x and y, or correlations along x and z, or correlations along y and z. I could have correlations anywhere in the 3-dimensional space. And now you need to start abstracting that to, say, a 10,000 dimensional space. Now this is where most people can't quite picture a 10,000 dimensional space in their minds. I certainly can't picture a 10,000 dimensional space, but you can imagine that it might exist. Okay, so if we have a 100 by 100 image, we have 10,000 pixels. That means that each image corresponds to a single 10,000 dimensional vector. If I have 1,000 images, I now have 1,000 10,000 dimensional vectors. Now, if I want to do principal component analysis, if I have 10,000 images and each one of them has 10,000 components, I can do this process called principal component analysis, and I can find the most important axes, the ones that have the strongest correlations in this 10,000 dimensional space, and then I can sort of rotate into an orientation where, say, the x-axis has very strong correlation, and the y-axis has the next strongest correlation, and the z-axis has the next strongest correlation, and gradually the correlations are getting weaker and weaker until we're left with only noise. Okay, so again, remember that we have this arrangement of n times n minus 1 over 2, so if I have a 10,000 dimensional space, the number of potential rotations I could perform in that 10,000 dimensional space is 10,000 times 10,000 minus 1 divided by 2, a very large number of different rotations. So this matrix that I'm talking about constructing, we're never actually going to write the whole matrix down. Okay? Now, if I don't have 10,000 images, if I only have 50 images, each of the images has 10,000 pixels still, but there's only 50 particles that I'm trying to classify or something, that actually limits the size of my output space. I only need 50 vectors to fully specify all 50 of my input images. So I don't need 10,000 basis vectors. I only need 50 basis vectors in this case. So the number of basis vectors that you would need to have to fully specify the system is the lesser of the number of images and the number of dimensions in the image. So if I had an image that was 4 by 4 pixels, and so it only had 16 values, and I had 100 of those, then I would only need 16 basis vectors to fully specify the system. If I have 10,000 pixel images, but I only have 100 of them, then I only need 100 basis vectors to specify it. So whichever of the numbers is smaller is the size of my output from this process. So we look back at this 2D refinement process again, and we're doing principal component analysis at this point. And principal component analysis is the process of looking at this 10,000 dimensional space and finding the strongest correlations. So you're basically looking at the correlations between every pixel and every other pixel in a sort of exhaustive way. It seems like it would be a really difficult mathematical problem to solve, and indeed there are a whole bunch of different algorithms to solve it, and in a lot of them take a long time to compute. There are some strategies, if we say I don't need to specify all 100 or 1,000 or 10,000 vectors, if I only need to find the strongest components, so I only want to find the 10 most significant vectors or the 20 most significant vectors, there are some very clever algorithms which can do this process very quickly, which can find the strongest correlation easily, and then it can find the next strongest correlation, and so on. Okay, so if you look at this process, when I'm doing my PCA here, there's kind of a problem. So I can do my PCA on these class averages, or I can do on these reference class averages, or I can do my PCA on the aligned particles. The trick is the particles have to be aligned, because what I'm looking at are variations. If I have 10,000 pixels, I'm looking at how well this pixel correlates with this pixel. If the particle is in this orientation in one image, and it's in this orientation in another image, this pixel no longer has very much meaning. So if you run principal component analysis on a set of particle data which just has completely random orientations in it, what you get out is something that looks kind of like circular harmonics. I mean, it's just kind of a very generic space where you just see patterns which are the same regardless of what the input data is. So we can only use this method on particles which have been pretty accurately aligned to one another, or aligned to some common set of references. So we need to have these self-consistent orientations. The problem is that don't have class averages when I start. How do I bootstrap this process? Where do I get the class averages that I use the first time I run the algorithm to do the alignment of the particles? If I just run PCA on the unaligned particles, I don't get very useful classification out. So I need to have some process I can use at the very beginning to generate some quick class averages which will allow me to do some alignment and then allow me to start using my PCA in a useful way. So the strategy we use to accomplish this is rotational translational invariance. So if you think back the other day when we were talking about Fourier transforms, you'll remember, we'll go ahead and do this again very quickly. Oh no. Wrong folder. Yeah, there we go. Okay, so hopefully you all remember this little example where we looked at different shapes and their Fourier transforms, and we put up this square impulse function here. Oh, I don't have enough sine waves, hold on. Okay, so we put up the square impulse function here, and if you remember, I said that we look at the Fourier transform of that square impulse function is just the intensity of these sliders over here on the left. And we have our phase information over here. But then I showed you, if we looked at a translated square impulse function over here in the box, that these sliders didn't change at all. So the amplitudes of the Fourier transform don't change when a shift occurs. This happens in 2 dimensions as well. So if I have a particle in a box and I translate the particle in the box, the amplitudes of the power spectrum, the Fourier amplitudes don't change. They're still the same. So that means that those values, the power spectrum of the image, is a translational invariant. It does not change as the particle translates. Okay? Now if we want to classify these particles, unfortunately we can't just have translational invariance, we also need to have rotational invariance. Now keep in mind what we're talking about here isn't a one-dimensional curve, we're talking about 2-dimensional images, so we have a 2-dimensional Fourier transform. So all I need to do is integrate out that rotational component. So if I take the 2-dimensional power spectrum and I do a rotational average of it, and I get my 1-dimensional power spectrum, that 1-dimensional power spectrum should be exactly the same if the particle is in the middle of the box or the edge of the box or rotated 50 degrees, whatever. No matter how I put the particle in the box, that power spectrum shouldn't change. Okay. So that means that I could take each of my particle images and I could compute the power spectrum for the 1-dimensional power spectrum for each of those images, and all of the points in my power spectrum would then become numbers which I could use as invariants, and I could use those to try and classify the particles. Now, in reality, we don't use the power spectrum. The power spectrum is kind of a stupid set of invariants. It's a simple example that's easy to conceptualize, but there are much better invariants that we can come up with which better represent the information in the particles. We throw away a lot when we compute a power spectrum. So we come up with a set of invariants for the particles. I can actually show you what they look like very quickly, and I'm just about done. And we'll look at this in a little more detail this afternoon, hopefully in the tutorial session. So when we did these 2D refinements, there are all of these files in here. One of the files in here are, let's see, so we have input FP. Yeah, there we go. So, so when it bootstraps this 2D class averaging process, it does it by computing these rotational translational invariants for a subset of the particles. So the particles look like this. So these were all the particles that it used to generate this initial set of class averages, and the invariants for those particles look like this. They're not very interesting to see. Let me zoom in a little bit so you can see what they kind of look like, but they have all these features inside. And then we can perform, instead of performing principal component analysis on the aligned particles, we can perform principal component analysis on the invariants, and that will allow us to then classify the particles just in this first iteration to give us some initial class averages. So the class averages we get out by classifying this way aren't going to be as good as the class averages we get out later, because when we compute these invariants, we have to throw away some information. We can't completely represent what's present in the images based on these invariants. We can just mostly represent what's present. So this gives us some initial class averages, and the initial class averages allow us to align the particles, and then we can get some better class averages out. Okay, I think that's... oh, I was going to show you one other thing, that's right. So one other thing that it saves when we do this 2D classification process are the basis vectors. So this is the results of that principal component analysis. So I look at the basis vectors. What I will see is some internal structure. The first basis vector is just basically the average of, well, or this is not actually basis vector. This is just the average of all of the particles. This first basis vector very often just looks like the average of all of the particles. It's the strongest set of variations that's present among the data. So what we're seeing here isn't an average of the images. What we're seeing here is a vector in this very high dimensional space defining where the strongest correlations between things are. So this is sort of indicating that there's a strong negative correlation among pixels here versus pixels and so on. It's telling us about correlations. So what we see is we look at higher and higher numbers. So this is the most significant information. This is the next most significant, next most significant. And gradually we get fewer and fewer of these components. And eventually this will just work its way down into noise. So we can select how many of these basis vectors we want to use when we classify the particles. These are very strong. These are very weak. So I could include more and more of these, but eventually I'm going to start including pure noise. And when we include pure noise, it makes the classification worse rather than better. So we usually just use a fairly small number of these basis vectors, like 10 or 12 or something like that. And it gives us a very nice classification of our particles into these coarse orientation-based groups. Okay, that is it. Any questions? I know that was a shock.
Thank you for an excellent talk. I'm sure there are questions. Yes.
I'm sorry I didn't explain how PCA works internally. It's just that's just one step too far.
Steve, with regards to your fatty acid synthase example,
yeah.
If you attempt a reconstruction of your multiple conformational states that are present, you focus completely on the side views which have the most prominent conformational state. Now, if you want to have the top and bottom views of the same particle, you won't be seeing much of a difference.
Oh, you'll see large differences in all the different orientations. You can do the same analysis I did in other orientations as well.
Okay.
So that particular data set has a fairly strongly preferred orientation. So I think we had something like 10,000 particles that were basically in that side view orientation.
Okay.
If we go to the top view orientation, we only had like 500 particles.
Right.
So you can still do the analysis. There's not as much signal there. It's just easier to see in that order. So, but that's purely 2D analysis, right? That's not when I say it's a top view. I'm just saying it looks the same. So I'm calling it a top view. I don't really know that that's 90 degrees with respect to that.
And probably a little unrelated to that, when you look at collecting data for particle classification for a certain molecule. Where do you stop? Like, is there an endpoint that...
You mean how much data?
How much data, for...
So, if you think about the example that we're doing in the tutorial this afternoon, we've got 5,000 particles, and that is going to get us to 3.6 angstroms. And if we have 20 or 30,000 particles, we get to 3.2 angstrom resolution. And it progressively gets harder and harder to make any more progress, right? At some point, you just hit a wall for really good homogeneous particle data. If the data is highly heterogeneous, like the fatty acid synthase, you have to define what your goal is. If you wanted to do a little dynamics movie like that in all different orientations, well, you'd want to collect enough data so you have 10,000 particles in all different orientations, and you can look at it in a very fine way. But there's no absolute rule. Basically, you should start by collecting, one session worth of data, whatever one session means to you, and process a chunk of that data and see where you are, and then you could decide whether you think you're going to make more progress by collecting more data or not. But until you have some initial set of data for a particular project, some projects only take 4,000 or 5,000 particles, some projects take 50,000 particles. It's very project dependent. Symmetry plays a role. I mean, if you have virus particle with 60-fold symmetry, then every particle is sort of the equivalent of 60 particles. So if you have 100, that's like having 6,000 ribosomes or something like that, right? That's just a general relationship. It's not an absolute rule. There will be some projects that need more and some that need less.
Thank you.
Yesterday, you briefly discussed about this GP160 trimer from HIV, the problematic
Yes,
case that
right.
material. So the problem came because using not down-filtered reference images for classification or for picking. I mean,
the problem occurred because the images were collected very, very close to focus, and the things that they boxed out of those images weren't real particles and didn't have enough signal to accurately do any alignments at all.
so it came from the supervised classification.
So they had references, and they'd used the references to do the particle picking, and they claimed to have proved that they had actually what they had picked out were actual particles. Now it's actually a very sad case because in some of the later work where they're trying to prove that they were right, they collected some more images on the same grids, and they did those far from focus, and they showed the far from focus images, look, there's beautiful particles all over the place. So we really did have particles present in there. And it's absolutely true. They did beautiful purification. There was nothing wrong with the biochemistry at all. It's just the guy who was using the microscope said, the best focus to use is Scherzer defocus, right? So he collected the particles at 0.2 microns or 0.3 microns, and you couldn't see anything. There wasn't enough contrast for any algorithm to pull anything out, and they had pure model bias in their results. And then they tried to prove that the last 10 years of cryo-EM history were wrong, and when we learned we shouldn't have model bias, they just said, yes, we should have model bias. It's not a problem. It's good.
I have a second question.
Yeah.
So could you, regarding this conformational flexibility, could you please comment on the cross-linking to arrest the conformation?
Well, cross-linking does reduce conformational variability, certainly, but then it's cross-linked. It reduces the biochemical relevance of the solution, obviously, but if that's the only way you can get things rigid, maybe it's not a bad strategy. So if I had a really dynamic system like that, maybe I would try and cross-link it and then solve one 3D structure in the presence of cross-links, and then do it again without the cross-links, and then try and relate that structure to the dynamics.
Steve, I have a difficult question.
Oh, good.
What's the difference between factor analysis and PCA?
You may be the only one who understands the answer to the question if I answer it. It's subtle. It's a subtle difference between factor analysis and PCA. In most situations, factor analysis and PCA produce pretty much equivalent results. In most situations, they produce pretty similar results. Not in all situations. It's not the same. I don't have a good pre-canned answer to do it without math. You have an answer. You want to tell me?
In general, factor analysis is used in medical researches, and very often it's related to the general features which are specific for this or this kind of
Yes.
so-called the predefined.
Yeah.
In principal component analysis, you are using nearly, objective parameters, according, variations and that.
I disagree with that point a little bit. You're right. Factor analysis can have provide priors. That's often used that way.
Overweighted. That's what the people, for example, perimeter of cells, etc. They're making an additional, weighting of the.
But what you're describing is one use of factor analysis. There are strategies for doing factor analysis without putting any initial bias on it as well, for finding the features directly from the data without forcing your viewpoint on it in the first place. And those methods, most cases, it produces another orthogonal basis, and it often produces very similar results.
Yes, it's a feature. Factor analysis is not always orthogonal.
It doesn't have to be, you're right, but it often is. Not always, you're right. That's why I didn't want to answer the question. I'm not aware of anyone in cryo-EM who's using factor analysis, are you? People have talked about it a little, but
Factor analysis very often used if you are talking of EM, in slices and very often in tissues.
Okay,
they do analyze
so for
example,
histology.
Yes.
Yeah, right.
Okay, you
right.
can install it.
Yeah,
But they're
right.
using quite often. And
Yeah.
when you're talking, for example, of the perimeter of cells or the square or etc., in this case, those those factors like contour or square of the area of nucleus of the cells, they're definitely cross-dependable. They're not orthogonal.
You're right. You're right. Okay.
Oh, well, thank you, Steve. I have one question, but maybe a very stupid question about your program. Since I want to understand this, you still have the translation movement and rotation movement to figure out the movement for the particles to align them before you to classify them.
Yes.
So, which kind of accuracy do you use for in EMAN2? Like, for sure, it's sub-pixel, right? Sub-pixel, smaller than. So,
it's up to you. If you looked at the program that we were using in the tutorial,
yeah.
there's a bunch of advanced parameters, and you can control all of those details if you like to. The default is to just do it with about 1 pixel accuracy, because we're really normally just after very low resolution features when we do this in EMAN. Since we're only using the class averages to make initial models and to make these little movies, we don't care if we're preserving very high resolution features in the class averages for that purpose. You can modify those parameters and tune it more for very sharp looking sort of class averages. It takes longer to run, and it doesn't really provide any benefit in the context that we're using it.
Okay. So on an addition on this, so for the algorithm you use for the shift translation alignment and the rotation alignment is the same, right? So, but I mean, operation.
There's one operation which does both translational and rotational alignments, and also usually a flip. But there are pieces in it where first it does the rotation and then it does the translation. I mean, it's
okay. So for sure that cross correlation, this kind of mathematical calculation will not work on this small.
Well, cross correlation can be used in certain contexts. If you filter the data in appropriate ways and stuff, you can still do cross correlation. Most of the translational alignment, if you've got the rotation dealt with, and you've dealt with the filtration properly, then you can use cross correlation to quickly find the translational alignment. Yes.
But yeah, you're right. But I was thinking so without translation alignment, probably we cannot do cross correlation, right? Otherwise, we cannot get good result. For example, the feature maybe it's not very round. Maybe it's for example, road-like shape, the feature.
Yeah.
So the other feature maybe on the other direction, road-like shape. So the correlation won't work.
Cross correlation won't work until the orientation has been dealt with. So
yeah.
EMAN has like 4 different algorithms that you can use to do those rotational translational alignments. One of them was developed by Paul Penchek, and that does an iterative. It does a rotational, and then it does a translational, and it does a rotational, it does a translational, and it goes through this iterative process until it converges. EMAN makes use of translational invariance to do the rotational alignment. I mean, the default algorithm that we use uses translational invariance to do the rotational alignment, and then it does the translation directly with some extra checking. But there are other algorithms too, exhaustive search, there's a local refinement to do subpixel accuracy, so it's not just one choice, it depends.
Okay.
We make choices for you, which we think are the best way of doing it, but you can always pick other ones if you like. I usually won't have much effect.
Thank you.
Yeah.
Hey Steve, excellent talk. I just have a question regarding the orientation of the particle in ice.
Yeah.
So we had some samples where we see that samples prefer to have a particular orientation in the ice. Have you heard about it?
Preferred orientation?
Yeah.
Yeah.
In the ice.
Yeah.
They have particular orientation. So when we distributed the density, when we see the density orientation, we see that there is a part missing.
Yeah.
In the thread.
Yeah. No, that's very common. So if you have continuous carbon, the particles tend to stick to the carbon when you're using it, and they will often stick in particular orientations. If you don't have continuous carbon, then you often find them sticking to the air-water interface and having a preferred. It depends completely on the particle and how hydrophobic it is and how much it likes carbon. So ribosomes, when they're done on continuous carbon, there's 2 different orientations which they tend to form, and you'll see a lot of the ribosome structures in the EM data bank were filtered in such a way that you can still see some of the smearing due to this anisotropic resolution that you get out when that happens. GROEL tends to be found in the top view and the side view and very little in between because it's got these hydrophobic patches on the surface. There are ways of experimentally getting around that. Part of specimen preparation, there are tricks people use to try and make the particles have various rotations. Are you telling me to answer the question, or do you want to answer the question? Okay, we're going to hear about that very shortly, so I'll stop answering.
Thanks, Linus.
Yeah.
During unsupervised mapping, what is the optimal number of selection of particles or images?
What's the what? Sorry, I missed that.
During unsupervised mapping.
Yes.
So what is the optimal number of selection of orientation?
There isn't one.
Or we can randomly select n number of, and then we can do the PCA and come to that.
So in the earliest versions of that algorithm, we actually used all of the class averages as alignment references. So the idea is you get the class averages and then you align the class averages to each other to get them in sort of a self-consistent set of orientations, and then you can align the particles to all of the class averages. It turns out that that's a lot of wasted computational effort. You can achieve basically the same results with a subset. But how many you need to have in the subset depends on how different the different orientations look from one another. For every particle, you want to have at least one of the references which looks enough like it that you get a self-consistent orientation out. So there's no magic number. Was I answering the right question? You look like I wasn't.
So how will you do for the spherical looking, exact spherical looking molecule?
Like, you mean like a virus, or do you mean?
Viruses.
Okay, I mean it has to have some features or there's nothing to align and classify, right? But for a virus, obviously you don't need very many references to it. I mean, you could probably make do with one or two because it's going to align based on the outside of the spherical shell. But if you have something like a ribosome, which has 3 different lengths, in the different elliptical axes, then you need to cover all of those different possibilities pretty well so it can align. Otherwise, you'll find it aligning to one edge or to the other edge, and you get all sorts of funny effects, and it doesn't work as well.
Steve, an excellent talk. Is it required to scale the particles or images before averaging?
Scale them?
Scale them. They bring to same.
Yes. When we did the demonstration, we downsampled the particles. You don't have to do that. You can do it on the full resolution particles. It just takes a lot longer. So most of the algorithms that we use in image processing in cryo-EM scale very poorly with size. Some of them scale as poorly as n to the 4th or n to the 5th power. So if you double the size, you're talking about taking 30 or 40 times more computation to complete. So we usually try and sort of optimally downsample the data to achieve whatever results we need to achieve at a given point in time. So that's why we now provide these different variants of downsampled data. We have the stuff designed for low resolution work and stuff designed for intermediate and stuff designed for high, and you only use the highest resolution data once you've got all the preliminary computations out of the way, because it's just a wasted effort.
Okay, thank you. If I understand correctly, do we need to go unsupervised classification initially and then go supervised classification later on?
If you know what your molecule looks like already and you have a good starting model, so say you're doing a ribosome and it's a slightly different ribosome, but it's still a ribosome, you could just start with a ribosome as an initial model and you could go directly to 3D refinement and skip all that initial model generation and stuff like that. If you're doing something you've never seen before, you don't know what it looks like, you don't know how much dynamics it has, then you certainly want to do some 2D analysis before you jump into 3D so you know what's going on.
Okay, and if we do things which is very new and we don't know anything about it, so does the defocus makes any differences?
Well, I mean, defocus always makes a difference. If you need to use a high enough defocus that you can see the particles. You don't want to put yourself in a realm where you can say, I think I see the particles, I'm just going to box them out and I think it'll probably sort itself out. That's not a good situation to be in when you don't know what you're looking for. Okay. If you're looking at a ribosome, then if you can barely see it, it may still be okay. As long as there's enough information that it can do the alignment, you'll know if you've got the right answer or if you have a problem, right? But if you're working on something brand new, you want to have enough contrast that you can kind of see by eye that at least you can be sure that the things you're picking out are actually particles.
Thank you.
Right?
Any other questions?
Hello, sir.
Yeah.
Sometimes when we manually pick the particles, then we are tend to pick some wrong particles also. So can we that like we sometimes we are biased to
I understand.
pick the
Yes.
particle.
Right.
So can we remove the bias by doing a multiple refinement or adding some noise to the data?
Adding some noise.
Yeah, like final data. Can we add some noise to remove biasness?
I don't understand how adding noise removes bias.
Like when you get the initial model, yesterday we did the initial model in practical course. Like initial model, when you got on the based on the initial model, then we again did the refinement.
Yeah.
Then if you do the like we did only one refinement, can we do the multiple refinement with adding noise to the initial model, so that you can remove some biasness?
Adding noise isn't going to be useful, I think. Yeah.
If you take the question is that if you have an initial model, for example, you have a new ribosome, human ribosomes,
Yes.
but you have to start from 70S ribosomes.
Yeah.
How to avoid the
bias of that
bias
initial
to
structure?
that the guy suggests to add the noise.
I understand what you're saying. You're talking about in the refinement process, the initial model, you want to filter it in some way to reduce. Yeah. Okay, so that's a standard part of most refinements nowadays. So this sort of gold standard procedure that people are using nowadays, when you take your initial model and you put it into the refinement, you say, I'm targeting 10 angstrom resolution. EMAN will automatically take your initial model and randomize all of the phase information past, say, 15 angstrom resolution or 20 angstrom resolution. There's thresholds for different values. So it'll go to something substantially worse than the resolution you're targeting, and it will make sure that that initial model has pure noise beyond that resolution. And then when you do your refinement, you know that it's not being caused by model bias. In Relion, they also do something, equivalent gold standard, but in that case, they don't add noise to the structure. They just go to, say, 60 angstroms, and they remove all of the information beyond 60 angstroms. So there's still a little bit of bias in that process, in that the even and the odd halves of the data. So we do these split. I said we would talk today, this afternoon, about the even and the odd outputs that we have in the refinement process. Right? So what you're doing is two independent refinements with the even half of the data and the odd half of the data. So in EMAN, the even and the odd starting models are completely different beyond some target resolution, because we've added noise, like you're saying. In Relion, they're the same, but they're zero. So it still seems to be okay. Initially, I was critical of that method, but I've tried it myself, and it actually seemed, it also seems to do an okay job of getting rid of model bias in most cases. Other software packages have other ways of handling the same problem. Nico Grigoriev absolutely hates this gold standard thing. He said, not a gold standard. So he's very critical of that, and he has his own strategies for avoiding model bias. When he computes the cross correlations to do the alignments, he actually takes the absolute value. Well, maybe he may not be doing that anymore. A method he used for a while was taking the absolute value of the cross correlations. So you could have a negative correlation or a positive correlation, and they would sort of cancel each other. Anyway, we don't need to get in that much detail. Each software package has some way of dealing with the issue.